Hi, everybody. It looks like I lost Amy somehow, so it will just be a minute. There we go. Sorry, I lost you somehow.
I have no idea. I was just sitting here. I didn't hear you. I assume you were speaking to others, but I'll just share screen again.
Yep.
And do you want me to just dive right in?
I have a little intro. We'll start with that, and then I'll hand it over to you real quick. All right, while Amy's setting up, welcome everybody. I'm really excited to introduce our speaker today, Dr. Amy Toth. Amy did her PhD with Gene Robinson, where she made key insights in and across several species. There she used gene expression data to make important contributions to our understanding of the evolution of maternal behavior, division of labor, and eusociality. She then completed a brief postdoc at Penn with Christina Grosinger before starting her own group at Iowa State, where she continues to study social insects, blending ecology, evolution, and behavior, some of our favorite things in this series. Though her contributions are broad, I would want to highlight one of my favorite bits of her work. Amy was actually a speaker at the first conference I ever went to, and it was the first time I had ever heard about Polistes social wasps. These queen wasps can chew up twigs to make tiny nests that we often see outside, and they will lay their eggs that they actually care for. So they bring food to their young multiple times and can build more and more complex cells into the nest and continue to grow their little family. In some species, there may be even multiple females that start the colony, but only one of them will produce the bulk of the offspring. And this delicate social structure is maintained partially by aggressive social interactions from the main foundress or queen. And these animals can actually use their faces to recognize each other, remember who they fought with, and gate their aggressiveness, which I just found fascinating. In this talk from now many years ago, Amy told us how she identified candidate genes involved in this fascinating behavior and used these insights to develop a hypothesis that led to her testing a pharmacological intervention that actually decreased aggression in these wasps. And since then, she's been a central figure in wasp evolution and the teams that have documented the worldwide distribution of social traits in wasps and the ecological pressures that influence cooperation, among many other insights that I'll let her tell you about today. And so with that, I just want to thank Amy both for introducing me to the wild world of social wasps and also for giving this talk today. Over to you, Amy.
Thank you so much, Emily. I really appreciate the invitation to speak to this crowd. And this online format is something new for me, but it's kind of fun and exciting. I know that there is another event occurring in an hour, and so I'm going to try to stick to time here. But I am going to give you a tour into the wonderful wild world of wasps today and tell you about some of our work on genomics of paper wasp behavior. And I actually will be visiting some of the topics that you just heard about, including some new data. So this is going to be a talk that is light on the neuro side because we really don't have a lot of neuro in these wasps yet. Pretty heavy on the ecology and evolution side, and then delving deeply into the genomics side. And so what I wanted to do is actually start with the world of wasps. And this is a wonderful photo by Alex Wild of a species of wasps called Seneca cyanea, found in Brazil. And you can see this colony of wasps that's attached to the side of a tree. You can see that they're multiple interacting individuals. These are all female, and they're members of a eusocial colony. There are all kinds of interesting things going on inside of that colony, including caste differences, dominance interactions. But then there are also many important things going on outside of the colony. So they're living in this complex abiotic environment that they need to respond to. They also need to avoid predators. They also need to avoid parasites. They also need to hunt for prey. They themselves are predators. And so we really need to be considering the environmental context in which the fascinating behaviors that these animals do occur. And I think wasps are just one example, but I think this theme runs true for all of us interested in understanding the mechanisms of behavior. And so one of the goals of my work has really been to integrate an understanding of the ecological and evolutionary context into our understanding of behavioral genetics. And so we know behavior is defined by being a response to the environment. So behavioral traits, I would say above and beyond other types of traits, by definition, are going to be highly responsive to the environment. That's just how behavior is. We know that many behavioral traits are underlain by complex gene by environment interactions. And we know that behavioral traits, by and large, are highly polygenic. So many, many different genes contributing to the regulation of a given behavioral trait. I'm just showing you one example here. This is work by Gene Robinson and colleagues, in which they have spent many years trying to understand the genetic basis of differences in behavior in honeybee workers. And these are gene networks that show that there are many interacting genes contributing to different types of social behavior in bee workers. For example, foragers, bees that take care of brood, known as nurse bees, bees that are aggressive and guard the hive. And we know that different but sometimes overlapping sets of genes are involved in the regulation of these behaviors. Now, if we want to try to pin down the association between a gene and a given behavior, that becomes a challenge. And that's a challenge because of these G by E interactions. And it's also a challenge because of the highly polygenic nature of these traits. But it's also a challenge because of the environmental context is so important. And so if we take the bee out of its hive, or we take the bee out of its ecological context, we actually may not be able to dissect the genetic basis of that trait appropriately. And so I'm coming at this sort of from a background of behavioral ecology. And in behavioral ecology, we're concerned with how behaviors allow an animal to adapt to its environment. And we're also interested in how the environment and changes in the environment influence behavioral decisions by individuals. So behavioral ecology is sort of one side that I'm coming from, but I'm also interested in functional genomics. And so those of us interested in functional genomics, we want to understand how genes regulate behavior. And of course, there can be feedback as well in behavior regulating how genes are expressed or used. So I think historically, there really has been kind of a divide between functional genomics and behavioral ecology. And so one of my life goals is to actually get past this and aim for a fuller integration of functional genomics and behavioral ecology. And this is what I'm calling ethological genomics. And this is an idea I've been developing recently with collaborator Mike Sheehan and postdoc Alex Walton. And the idea here is that we need to provide equal attention and weight to the genes underlying behaviors and the environment and their interactions. And that if we are to really fully understand gene behavior relationships, we need to fully understand the ecological and evolutionary context in which those behaviors occur. And we have this little cartoon here of a person walking across this bridge carrying a box full of tools. And one of the tools that we've been using heavily in the past few years and that I'll be talking a bit about today is RNA interference. And so we don't have mutants. We don't have a lot of tools in our toolbox because a lot of times when we're working in the realm of ethological genomics, we're not working on classical model organisms. And so our toolbox is somewhat limited, but we do have some tools. And I'll be telling you about some of the things that we've been doing in that realm today. So to define ethological genomics, this is a field of study involving the integration of behavior and genomics, the goal of which is to understand the molecular basis of complex traits in their ecological and evolutionary context. So I'm going to kind of go through each of these and why today I will argue that I think social wasps are a really good model system for ethological genomics. So social wasps, you have seen them before. They probably live on your house. And so this is one example. This is Polistes dominula. This is the European paper wasp. And they have really fascinating behaviors, which I'll be describing in much more detail today. But they live on these eusocial colonies shown here. They have these open combs of their nest, and you can walk right up to a colony and you can watch all of their behaviors. They're not hiding. They're out there in the open, and you can see their social interactions. You can individually mark them, and you can track how individuals are behaving with each other. And so Polistes is a genus of social wasps that are cosmopolitan and very commonly found. They love living on buildings, under the eaves of buildings. You have probably seen them. The genus name means city builder. And I think they're great models for ecological genomics because no matter where you live, you can find them. They have small tractable genomes, and I'll be giving you an example of that in a moment. They're relatively easy to rear and manipulate. We can't do continuous rearing yet, although it's something that we're working on. But you can bring them in the lab, and they readily survive and behave in the lab. And you can also work on them readily in the field, as I described before. You can walk up to a colony in its natural environment and watch what they're doing. And they have absolutely fascinating social behavior, which I'll be talking about today. So what I'd like to do today is describe some of our efforts in ethological genomics that have been supported over the past several years by a series of grants from the National Science Foundation and currently by the EDGE program for non-model functional genomics through the National Science Foundation. And I would like to acknowledge my collaborators on a lot of this work, Michael Sheehan and Elizabeth Tibbetts. Okay, so I'm going to go through why I think Polistes is good in all three respects: the molecular basis, the complex traits, and the ecological and evolutionary context. Okay, so first, the ecological and evolutionary context. These wasps are fascinating from an evolutionary perspective. So they are eusocial, which means they have cooperative colonies in which there is a single reproductive queen that is reproducing, and there are workers that never reproduce. And so this is known as a caste system, and that is the defining trait of eusociality. Wasps are eusocial, bees, some species are eusocial, and ants are eusocial. But fascinatingly, eusociality has evolved multiple times convergently across different insect groups. And the wasps, I think, are particularly interesting because today you can find a wide variety of different extant social forms, some of which are highly, highly social, like those Seneca wasps that I showed you in the first slide, some that are still solitary, and some that are kind of in between. And that's one of the reasons why I think Polistes are super fascinating and also really good because they kind of give us a window into some of the transitional stages in social evolution. So they have what we call primitive eusociality. So they are eusocial, they have queens and workers, but their colonies are pretty small. So compared to a colony of ants, which can have millions of individuals and several species, or honeybees, which can have tens of thousands of individuals, these colonies are small. So this colony that you're looking at here actually may only have a handful of individuals on it at its full mature size. The colonies also are annual, and so they start off actually with oftentimes a single wasp. She's the mom, she's doing everything, and she's founding the colony. We call these foundresses. Or she may team up with a sister or usually cousin, a close relative, and start that colony. And in that context, they actually have dominance interactions, which determine who gets to lay the eggs. But even when the colony becomes mature and we have a queen who is the only one who's laying the eggs, we have this colony where workers are present, and I can't tell you who the workers are in this colony. They are morphologically identical to the queens. Also, really key is that workers can mate and become queens. So unlike a honeybee or an ant colony where the workers have lost the ability to mate, these wasps actually retain the ability to be any caste at any time during their adult lives. And so for these reasons, these wasps have been important models for the evolution of cooperation. So a lot of the work, foundational work, like William Hamilton's work on kin selection, for example, was inspired by Polistes. And also because of this sort of intermediate sociality, which we can call primitive eusociality, they have been key for our understanding of caste evolution as well. And I'll be talking more about castes today. I need to get into the natural history just a little bit more and describe their colony cycle because it's highly relevant to some of the things that I'm going to be talking about today. And so Polistes in the temperate zone, like the species I'll be talking about today, have an annual colony cycle. If we start off in the spring, this is when colonies are founded, and this is typically done by these foundresses, which are females that have mated, and will initiate nests either by themselves or in small groups. Then they have a first set of brood, which are all female. Their daughters will typically become the workers on the colony. So in this early stage, we have both workers and queens now present on the colony. As the colony matures, these workers now take over all of the work, that is foraging for food, defending the nest, building the nest, whereas the foundress becomes a queen. She just focuses on laying eggs. They work together to raise more brood, which emerge later in the season, and this is what enters the reproductive phase. During this time, males finally are produced, which will mate with other queens and disperse. They don't work. But importantly, at this time, the colony also produces gines. You can think of these as princess wasps. So these are more likely to become queens the following year. And then finally, the colony enters its decline phase. The workers die, the old queen dies, and the only ones that survive are these gines, which will then overwinter. And then those that survive that overwintering will become foundresses the following spring. And we actually know a lot about the ecological factors that are important at the different stages of colony development. For example, nest founding failure, food shortage throughout the year, overwintering survival are big bottlenecks in their ecology and their nest survival. And we also know that this is a kinship-based structure in which cooperation is structured around kinship. So this is a mother-daughter cooperative society. Okay, as far as the molecular basis, this is something that we've been working pretty hard on for the past decade or so. About 10 years ago, there were 0 genomic resources for social wasps, and we've been working hard to change that. We started with a genome for Polistes dominula, which is a species that's native to Europe but invasive in the US and other parts of the New World. And this is the best studied Polistes species. This was work by former graduate student Daniel Standage. We actually sequenced the genome of a single haploid male. So these have this really interesting genetic system in which the males are haploid, but the females are diploid. So we had no genetic variation. We did Illumina sequencing. This was several years ago now. We got a really nice genome out of it. Over 12,000 gene models. We think most of the genome is represented in this. Since then, we've been doing a lot of additional work. We have whole brain transcriptome data. We have done population resequencing of individuals from native populations in Italy. And now currently, we're doing genome improvements. So we just recently got some long read PacBio data and high-seed data. So we're working now on chromosome level assemblies for this species. But we're not alone. We have some friends. So in addition to our Polistes dominula genome, there have actually been genomes released for 4 other Polistes species by other groups, including Polistes canadensis, and 3 North American species. This by Mike Sheehan's group just this past year. Okay. For me, the most exciting part of working with Polistes is their behavioral traits. And I think they have some of the most fascinating behavior, and that's why I study them. It's not because they're super easy to work with. It's because their behavior, to me, is completely fascinating. So not only do they have this eusocial system, but they have these castes being the defining feature of eusociality. So within a eusocial colony, we have a reproductive division of labor. And this is essentially your queen and worker caste. So the queen is the one that lays all the eggs, and the worker is the ones that do not lay eggs and do all the other tasks related to colony growth and maintenance. And I'm showing you examples from across the social insects here, showing the striking difference between the queens and the workers in many different species. We also have a division of labor between the subcastes of the workers. So in other words, not all the workers are doing the same behaviors at the same time. Let's say this bee might be tending the brood, whereas this bee might be foraging. And so they actually will specialize on different behavioral tasks. Now, from a genetic perspective, what's really interesting about this is that we see these huge differences in phenotype, but these are usually the result of environmental influences on gene expression, not heritable genetic differences. In other words, any female egg can grow up to be a queen or a worker. depending on the environment in which it develops. Or they could grow up to be, let's say, a soldier ant or a miner ant, depending on the environment in which they grow up. So the genome does not differ, but the gene regulation differs profoundly. So this is one of the reasons that we've focused strongly on gene expression differences and their role in generating social diversity in the social insects. So castes are one of the things that we focused on a lot. And in Polistes, we have even more dynamic situation. We have flexible castes. So we do have queens and workers, as I described before. They are eusocial, but every single female can become a queen. And she can actually become a queen at any point in her life. And that's what makes these castes especially flexible. So despite that, we do know something about how this process works. So when an egg is laid, as long as it's a female egg, it has the potential to become either a worker or a queen, but typically there is a caste bias during larval development. Larvae that receive less food early on in the colony cycle, maybe they're being raised by a single mom, they're not getting a whole lot of food, these tend to become workers. Whereas larvae that are raised later on in the colony cycle, they get fed more by a whole team of sisters, they tend to become queens. And so we call these worker destined or queen destined. However, when they emerge as adults, they still have some options open to them. So a worker actually can become either a subordinate worker, because there are dominance interactions among individuals on the nest, which determine their ovary size and subsequent reproductive capacity, or a dominant. And this is established through aggressive dominance interactions among individuals. And occasionally, and this is why the arrow is so thin here, dominant workers can take over as replacement queens, let's say if the queen mother dies unexpectedly. So it's not something that typically happens, but it is an option. We also have these queen destined larvae. Again, these are our princess wasps that are going to overwinter and likely become queens the following year. However, as I mentioned before, in the founding stage, they can team up with other foundresses, or they can go it alone. So they can either be solitary or they can engage in these foundress associations. When they do engage in these foundress associations, they actually have dominance interactions among them as well. One or more becomes subordinate, and one becomes the dominant egg layer. And that is the one that will become the queen when the colony matures. And again, this is all established through these dominance aggressive interactions on the colony. We also know that there are other complex social traits that accompany this really interesting social organization in wasps. And one that you just heard about that I am also going to talk about today is this amazing ability of wasps to recognize each other's faces. And this is only known in vertebrate societies, especially primates, and these wasps, incredibly. And this is work by Elizabeth Tibbets and Michael Sheehan, who I've collaborated with on some of this work. Elizabeth Tibbets actually discovered this as a grad student at Cornell, and has learned so many interesting things about it since. And so we've been working with them to try to understand what are the mechanisms underlying facial recognition. So you can see these wasps, they're all members of the same species, Polistes fasciatus, but they all have different faces. And that facial variation is something that the other wasps actually use in individual recognition. And because they have these vertebrate-like social dominance hierarchies, this has selected for social cognitive abilities in these wasps. And so you see these dominance interactions, such as the ones photographed here in this species, Polistes fasciatus, which is a species that actually has facial recognition abilities. And they use this in order to, well, not keep fighting with the same wasp over and over again when they meet in subsequent encounters. And I'll be talking a little bit more about that later. So what I'd like to do now is go through, now that you have a little bit of background on the system and why we think it's useful, to give you some case studies on how we've been addressing ethological genomics in social wasps. And the first one relates to social caste. So what we wanted to do here is try to understand how caste differences are set up. And we did this by first focusing on this caste-biased larval development. In other words, we went out in the field, we found nests in which they were rearing worker-destined larvae, those early season larvae, and we went back later and also collected nests from the field in which they were rearing queen-destined larvae later on in the season. and then we subjected them to transcriptomic analysis using RNA-seq. And this is the work of a former grad student, Allie Behrens. And this was with a native North American species, Polistes metricus. And Allie found almost 700 genes which differed in gene expression between these worker-destined versus these queen-destined larvae. We also knew that the nutritional profile of these wasps are very different. So we did a second experiment in which we looked at the role of nourishment in setting up these gene expression differences. And this helped us hone in on candidate genes that we thought actually would be more directly related to caste rather than related to something else such as seasonality. And so we compared wasps that had high versus low diet and found 250 differentially expressed genes. And then we looked at those that overlapped between these two RNA-seq studies. And this helped us identify best candidate genes for caste bias development. And these included a lot of different genes. Some of the ones that I wanted to highlight today, there were several genes involved in metabolic processes, including oxidation reduction activity. And the one that I'll be following up on today is NADH dehydrogenase, which we think is probably involved in regulating caste-related differences in growth rate, basal metabolism, and possibly brain development between the castes. So we subsequently wanted to follow up on this and do some functional genomics. But first, we had to make sure that our caste biomarker genes were actually going to give us consistent caste-biased expression. So what we did is we confirmed this separately using real-time PCR and indeed found that the worker-reared larvae had higher expression of several caste biomarker genes, including this NADH dehydrogenase gene. And some work by former postdoc Jenny Yunt identified several other very robust caste-biased genes, including inositol oxygenase, a metabolism gene, and heat-shock protein 90A, a stress response gene. However, we wanted to do our functional genomic studies in the lab. So we can bring these wasps in the lab, and here's just some neat pictures of how we rear them in the lab. We can put them in little plastic boxes. We can give them colored construction paper where they build nests, which they build nests with. We can paint mark them and observe their behavioral responses and feed them and watch their foraging behavior. So we were hoping to be able to manipulate some of these caste biomarker genes in the lab and study how they, when we manipulated those genes, how that affected caste-related traits. However, we found something quite disturbing, that lab life totally changed everything about their biology. And so again, Jenny Yunt, a former postdoc, found that when you bring wasps in the lab, it leads them to become different physiologically, behaviorally, and with their gene expression. So they become much fatter when you rear them in the lab compared to the field. They actually become very lazy. So this is the number of nest cells added. The nest just doesn't grow, and it's actually quite obvious that they're not really functioning normally when you bring them into the lab. Even though they're alive and thriving, they're not being active. And these caste-related genes that we were so interested in studying showed expression that was completely opposite of what we expected. In other words, workers showed queen-like expression of caste biomarker genes. So if we wanted to manipulate these and do functional genomic studies, we have a problem with doing this in the lab. So what did we do? We did it the hard way. We went and did it in the field. So this is some heroic work by a former grad student, Amy Geffrey, and a former postdoc, Susan Weiner, who used RNA interference in the field. And they went out to Polistes nests, which we reared in these little wooden nest boxes out in the field. They were free flying. And they actually fed double-stranded RNA for our candidate gene, NADH dehydrogenase, to larvae that were on the trajectory to become queens. And so they hand-fed these right into their little mouths. And what they found was a knockdown of the target gene, and that this actually led to an increase in one phenotype. Now, we wanted to look at caste-related behavior, and what we managed to get were data on lipids. Now, lipids we found were higher in the knockdown, and lipids were lower in the knockdown as compared to the GFP control. And this is a worker-like trait. In other words, wasps that were on the trajectory to become queens, when we treated them with the RNAi, became more worker-like in terms of their lipid stores. Now, what we really wanted to know was what happened to their behavior. Unfortunately, an evil raccoon came along and ate our experiment, and we were never able to follow up and look at the adult behavior of these wasps. So this is something that actually we're doing currently right now. So the take-homes from this first case study is that so far we found that Cas differences in wasps are associated with large differences in developmental gene expression. We found several hundred genes differentially expressed. The expression of these Cas-related genes is environmentally sensitive, and we think bringing them into the lab results in changes in food level and activity level, and that we basically had to study them in the field. We also found that NADH dehydrogenase seems to be a reasonable candidate gene for Cas-biased development. As far as the behavior, we'll see. That's something that we're still currently working on. And I think this is a good proof of concept that RNAi is a useful tool for functional genomics in non-model species. It can even be administered in the field since we're not doing any germline manipulations, so these can be free-flying out in the field. But there are certainly challenges, least of which are hungry raccoons. Okay, so that's our first case study. The next one I wanted to talk about was the dominance behavior of these wasps. So, as I mentioned before, these wasps engage in near constant fights with each other. And this happens throughout the colony cycle. Pretty much all the females are doing this all the time at some level. This can occur between foundresses, this can occur between workers, and also the queen is constantly dominating her daughter workers. So dominance is key to how these societies are regulated. So among adults, we have dominance between workers here and between foundresses. So what we wanted to do is actually look at the gene expression patterns associated with dominant and subordinate phenotypes and look at these actually across these different castes as well. So to do that, I'm going to show you the results of a somewhat old now transcriptome study in which we looked at the brains, the whole brains of individual wasps that were either dominant or subordinate. So we had dominant foundresses and subordinate foundresses, which we determined their status looking at their ovary size. We had active egg-laying queens, and then we had dominant and subordinate workers, again, determined by dissecting their ovaries. We found 500 brain differential expressed transcripts, 57 of which tracked dominance. And this is just a table here showing some of the example genes associated with dominance. Included are some interesting ones, actually, that have been shown to be associated with aggression in Drosophila, such as ebony and black. Some genes associated with metabolism of biogenic amines, such as acetylcholinesterase, and some that seem to be stress response genes, such as heat shock factors. Overall, we found that brain gene expression was associated with the colony cycle or social environment. In other words, the foundresses resembled each other, and then the queens and workers, again, they're on the colony at the same time, resembled each other overall in their expression patterns. So this tells us that there are really potent effects of the social environment on overall brain gene expression. And this, I think, serves as a red flag that we really must consider the social environmental effects on brain gene expression in this species. So one of the candidate genes that emerged from this study was actually a bit of a surprise. And this gene was vitellogenin. Vitellogenin is a yolk protein, and it's found across insects and is usually thought of as something that's being produced heavily in the reproductive tract of females. However, vitellogenin is actually also behaviorally active and has been shown to be in a wide variety of different social insects. And so when we looked across our study, as well as other studies that had looked at dominance in other types of social insects, such as bees and ants, vitellogenin actually turned out to be the single best predictor across species of being a dominant egg layer. And yet it's in their heads. So it's not in their ovaries. Remember, we're talking about brain gene expression again. So subsequent studies on honeybees looking at the localization of vitellogenin expression actually found that vitellogenin is expressed in the fat body that lies adjacent to the brain. And it's possible that vitellogenin may be communicating the physiological or reproductive status to the brain, because subsequent research in honeybees actually found the presence of vitellogenin receptors in the brain, suggesting that this could be one mechanism by which vitellogenin, as an indicator of reproductive status, could be influencing brain activity. And this is just a graph from a study that I did collaboratively with Fabio Manfredini, showing that vitellogenin is highest in the most reproductive and dominant females. So these are dominant and single foundresses, having higher expression of vitellogenin in this head fat body compared to subordinates and workers. So this turned out to be our single best predictor of dominant status. So what we wanted to do is then test for causality. And this is some work, very recent work by Alex Walton, who is a current postdoc in the lab. And Alex used vitellogenin knockdown using RNA interference again. And he did this in the laboratory. And he was able to get a knockdown through injection of double-stranded RNA into the heads of adult paper wasps. And so he did this this knockdown. He found that there was a significant knockdown in dissected brains. Again, likely this is in the fat body tissue surrounding the brain. And then he did dominance assays to look at whether the behavior of the wasps that had received this injection was altered in the laboratory. And he looked at both foundress pairings in the lab and workers. So what you can do is take two wasps, put them in a petri dish, and if they've never met before, they will fight. And you can score the amount of aggressive behavior. And what he found is that those that had received the vitellogenin knockdown actually had lower levels of aggressive behavior as opposed to those in the control GFP group. However, interestingly, he found cast differential effects. So he found no knockdown or behavioral effect in workers, only in foundresses. And we're not totally sure why, but it might have something to do with baseline levels of vitellogenin just being lower in workers. And so we're not getting an efficient knockdown there. So the take homes from case study 2, we found that dominance behavior is associated with differential expression of hundreds of genes. Our data suggests that the gene regulatory basis of dominance may depend on caste and social environment. And we've identified vitellogenin as a candidate regulator of dominance behavior in social insects. And I say a master regulator because of its demonstrated role across a wide variety of species. And we also know that it interacts with a lot of other systems in social insects, including juvenile hormone, which we know to be a major regulator of behavior and physiology. Okay, the last case study that I want to talk about is related to facial recognition. So this is a really neat behavior that is very specific to paper wasps. And interestingly, in the context of their dominance interactions, individuals need to recognize each other. However, it varies across species. And so what you're looking at here are photographs of two different species of Polistes. And Polistes fuscatus on the top is a North American species which has variable facial color patterns. You can see A and B are very different from each other. Whereas Polistes metricus, another native species that we can find here in North America, does not have any differences in its facial color patterns. And so extensive work by Elizabeth Tibbetts, Michael Sheehan and colleagues has demonstrated that Polistes fuscatus specifically has specialized learning abilities for faces, and that this appears to facilitate recognition of individuals during the founding stage. That is when they're trying to figure out whether to associate with another wasp during that founding stage or go it alone. And it also seems to be associated with reduced aggression towards familiar opponents. So if they've already met, they can use the face to recognize that they've already met, and then they tend to be less aggressive in subsequent encounters. And this is probably a way to save energy and the costs of fighting. So this is really a neat system because not only do we have this facial recognition, but Polistes metricus is another species which we can use as a contrast, which does not have facial pattern variation. And although they can learn to recognize faces, they're not very good at it. In other words, they're not any better at learning faces than other types of visual pattern information. So it does not appear to be a cognitive specialization, whereas it is in Polistes fuscatus. And again, this is foundational work by Tibbetts and Sheehan. So I teamed up with Elizabeth Tibbetts and former graduate student Allie Behrens to try to understand some of the mechanisms underlying this behavior. And we did a transcriptomic study in which we used this two-species system to try to pinpoint which genes might be associated with facial recognition abilities. So we compared Polistes fuscatus and Polistes metricus that were either trained to patterns or trained to faces. And we did this using a Y maze in which individuals were given a choice and they were given two different images and they needed to choose appropriately to avoid electric shock. And we then could study face specialization in Polistes fuscatus. So you can see that they are better at correctly identifying faces compared to patterns. That's this A versus B here. Whereas in Polistes metricus, well, yes, they're better than 50%. So they can learn faces, but they're not learning it any better than other patterns. In fact, they seem to be a little bit worse at it. So in Polistes metricus, we're studying just generalized visual learning, whereas in Polistes fuscatus, we can study this face specialization. So what we did is we trained these wasps. Actually, Liz Tibbetts did this part of it, trained these wasps, harvested the brains, and then looked at patterns of brain-wide gene expression using RNA sequencing. And what we found were 237 different transcripts associated with face specialization in Polistes fuscatus. So this is just a heat map showing the different gene expression patterns. We found that 37% of genes were upregulated in face trained versus pattern trained. And these included many different molecular functions, including ion binding, G protein coupled receptor activity, oxidoreductase activity. That's what I'm showing you down here is just a graphic showing you the molecular functions associated with these 237 genes using a gene ontology analysis. And some of the candidate genes that came out of this included serotonin receptor and tachykinin. So this was an interesting study to give us some initial hints as to what might be going on in the brains of these Polistes wasps that have this specialized form of learning. Next, what we wanted to do is try to get a little bit deeper into trying to understand how these facial memories are formed. And so to do this, we chose specific candidate genes that we thought might be interesting based on their previously understood roles in Drosophila learning and memory or visual learning. And what we did was we staged encounters between Polistes fuscatus, this is the species that recognizes faces, in the lab. And then we allowed wasps to interact multiple times. And then we would dissect the brains of wasps that were either alone, wasps in their first meeting, that is when they were just first forming the memory, and then wasps in subsequent meetings, in other words, wasps that were recalling that facial memory. We observed and measured aggressive behavior, and then we expected to see a decline in repeated meetings if the memory of others is occurring. In other words, once they meet each other again, their aggression usually decreases. And then we examined the expression of candidate genes using real-time PCR on these candidate genes. And what this resulted in was 3 candidate genes, NCKX30C, IP3K, and IP3R, which have become top candidates for memory recall because they showed unique expression during that second meeting compared to the first meeting or wasps that were housed alone, suggesting this was actually the memory recall part of it. These 3 genes all had roles related to calcium signaling in Drosophila, so we thought that was a particularly interesting line to follow up on. So then, again, we don't have a whole lot of tools at our disposal, but this time we wanted to use a pharmacological approach. And we decided to use a lithium chloride treatment, which was previously shown to alter NCKX30C and IP3K expression in Drosophila and to result in alterations in calcium signaling. So we fed wasps lithium, essentially. And what Allie found, Allie Behrens, again, the graduate student who did this work, she found that the lithium chloride treatment may enhance social memory. So here you have the controls, the vehicle, and then a non-target treatment, which was magnesium chloride, which didn't show any decrease in aggressive behavior over first, second, and third meetings, whereas the target treatment with the lithium chloride did show a significant decrease in aggression with subsequent meetings. And so this suggests a possible role for calcium signaling genes in facial recognition, although admittedly, this pharmacological manipulation is a bit of a blunt tool. And so what we'd like to do moving forward is do some more targeted manipulations of gene function. So the take homes from case study 3 is that we found that facial recognition as a cognitive specialization appears to be associated with specific genes that are distinct from general visual learning. So the genes that we found for the non-face learning species were overall quite different than those for the face learning species. We also found potential candidate genes related to calcium signaling for facial memory recall. And we used a pharmacological approach to give some support to the possible roles of these genes. However, this is a bit of a blunt tool, but one that can be useful for getting at causation for non-model ethological genomics. So with that, I'd like to summarize and end here. So I think I've shown you, hopefully, that we can identify candidate genes for complex ecologically relevant behavioral traits, and Polistes is a great model system to do this. I've given you some examples from CAS development, dominance behavior, and facial recognition of how we can accomplish this, and some of the candidate genes that we have generated from this work. We've also been really excited about RNAi as a useful tool for probing gene behavior relationships. And what I am most excited about this is that we can use it in a realistic or naturalistic context. We can bring this out in the field. We can actually do this right in the environment of ecological relevance. Also, I hope that you are somewhat intrigued about the idea of using non-model systems, that I think they can provide unique opportunities for understanding complex traits that may be somewhat specific, like facial learning, hard to study in vertebrates, but perhaps there may be parallels in invertebrates that might be more amenable to manipulation. And finally, my real take home is that gene function must be understood within its environmental context. So we would have made some grave mistakes if we were not thinking about the social context of gene expression and the environmental context, that is lab rearing, perturbing the function of genes. If we did not consider that, we would have big problems in understanding gene function. And so the social, biotic, and abiotic environment can have profound effects on gene expression, and thus our interpretations of gene function. We need to keep this in mind as we move forward. So just to mention really briefly a couple of the future directions with my work, I'm currently working on two fairly big projects. One is a broader comparative study, working with this great team in which we're actually doing 16 bee and wasp genomes and trying to do more of a comparative evolutionary study of how social behavior evolves on a molecular level. I also have a grant with Michael Sheehan in which we're trying to understand, develop functional tools for doing more genomic work, including CRISPR-Cas9 and hoping to do some single-cell RNA-seq to get at more of the neural populations that might be involved in some of these behaviors. And if you are really intrigued, ask me about our WASP Squad Lab Exchange Fund. So as part of this NSF Edge grant, we actually have money to give away to people who might want to try working on wasps. And so we're open to collaborations if you are feeling brave about working with a non-model system. Okay, with that, I'd like to thank all the people who did the work, my wonderful collaborators at Iowa State and beyond, many former and current lab members, including a great number of undergraduates who've helped with this work. If you want to know more, you're welcome to visit my website, email me, or follow me on Twitter. And this is our hashtag WaspLove, because wasps don't get enough love, and they are really fascinating and wonderful creatures. So with that, I am happy to take any questions.
Thanks. That was really fun. I can start us off. It usually takes people a few minutes to type things out. So I was wondering, for the vitellogenin, I'm still surprised always that that is one of the best candidates. And I was wondering if you'd looked at if there are different maybe isoforms that differentiate kind of brain vitellogenin and like the more classical roles that maybe we're familiar with from the literature.
Yeah, that's a really great question, and we haven't looked yet, but I think that's entirely possible, and that could partially explain what might be going on as well. That there could be, something that's more brain specific that might be cross-communicating the reproductive status of the wasp to the brain more directly. But we need to dive more into that, and that's one of the things that we hope to do moving forward.
And I was excited about the like single cell RNA-seq slide at the very end. I was wondering if you're also considering like maybe dissecting the brains to see if different regions give you better results, or using some sort of like early activity kind of markers, like like we would use CFOS to try to figure out like which parts of the brain maybe you could focus on to get stronger like signals from the RNA-seq.
Yeah, for sure. Yeah, and I think that's something that we have done some brain region work, and I think moving forward, there are opportunities to do laser capture and look more targeted at regions or even cell populations within the brain. And so I think that, we're kind of starting from zero. And so we decided to do whole brains, partially for logistical reasons, but also partially just to start describing where to start and get some leads. And so I think moving forward, I think that's really the direction that we need to go if we really want to better understand the brain regulation of these traits and better understand what's going on at a neurobiological level, which is something that, as I mentioned in my initial slides, like we're really kind of in the infant stages with this species as far as the neurobiology.
And I have one more just kind of very broad ignorance question. Like I live in New Jersey right now, and it gets pretty cold here. But every year we have paper wasps. And you mentioned there's this overwintering, but I was just kind of curious what that is.
Yeah.
Like, what do they actually do? Do they dig into the ground? Are they just really frost resistant?
Yeah. So they do overwinter as adults. So it's just the gines, and those are those princess wasps, and they will usually go under like tree bark, or your attic is a favorite place. Like finding crevices or places that are somewhat protected from deep freeze. But they will go into diapause, and so they pretty much shut down their physiology. Their metabolism is extremely low, and they just wait it out. And then, when spring gets going again, they've been metabolizing their fat stores to kind of survive on that, and then get going up again in the spring. But yeah, I mean, it's usually they're not getting frozen solid, but they're trying to stay protected from getting totally frozen. And so that's through like habitat selection in the fall when they're going into overwintering. They also overwinter in groups. So they will actually form big piles of wasps. So if you get really lucky, you can go in the attic and maybe find like a big pile of overwintering wasps. And that actually seems to help them be protected from freezing in the winter as well, forming these groups. And they'll even form fairly large groups, even with non-nest mates. It's not something where they're like being really picky about who they overwinter with. They just kind of cluster together.
I see. And I also wondered if there's any like, so I have to admit, I haven't read some of these papers in a long time. So you may have already answered this in some of your work, but so I know you looked a lot at the ecological kind of environment that leads sometimes there's more of these like cooperative groups or more solitary female nests. I'm wondering if you've seen like diet specialization or like they're predators, right? So
yeah,
like, do you see other differences that kind of correlate with that, or is that just like a basically impossible question because they fly?
Yeah. So do you mean within a species or across species?
Either.
Yeah. Yeah. So we don't really know that much. They're pretty generalist in their foraging habits. They do forage on flowers as well, so they are pollinators, and mostly foraging for nectar, not pollen. But then they're preying on insect prey, so their prey are usually soft-bodied caterpillars and things like that. And we don't know if there are some differences between species or between nests and how they're dividing up the resource space as far as the different diets that they might have. But it's very possible. And that's actually one of the things that between Polistes fuscatus and metricus, they're kind of a mystery, because I didn't get into it too much, but these two species are fairly closely related. They live in the same place, so they're not geographically isolated. And so there's been this, speciation event several million years ago, probably, and we don't really understand how they're dividing up that niche space exactly, because they seem to be filling very similar ecological niches. And so it's not, one species is a little bit bigger than the other, and so it's possible that maybe the prey size variation might not overlap that much because of the size difference. We don't really know.
That's really exciting. Okay, so with that, I think we will wrap up. Thanks so much for coming, giving this talk. It's been lovely to see you, and good luck.
Good luck to you too, and thank you so much for the invitation. And I'm happy to take questions if anybody wants to email me.